So as we've seen with some of our functional groups, not only can they be present in straight chain structures like we saw in the previous slide, some can also be present in ring structures, and this is also true for the ether functional group. So here we can see ring structures that contain the oxygen atom and therefore have the ether functional group. So on either side of our oxygen, there's actually an attachment to carbon. Remember, each position in this ring structure that isn't shown as an atom type is presumed to be carbon. So we've got the oxygen attaching to one carbon on this side and one carbon on this side. So the R group is what it's attaching to on either side here. What makes it a ring is that each R group actually ends up connecting to the other one to form this whole ring. So the rings that have 5 atoms, 1 oxygen and then 4 carbons, are called furan, and this hexagonal ring is called a pyran ring. Don't worry too much about these names. Just want you to know that the ether functional group can be present in ring structures, and we're going to see this quite a bit because this is what we see in sugar unit structures like glucose and fructose that are in this cyclic form. They all have the ether functional group within their structure, which is what we can see here in the next part of the slide. So here is the ring structure of glucose, and we can see that it's pointing to that ether functional group where we've got an oxygen atom and it's attached to carbon atoms within this ring structure. So we can see that the fructose forms a 5-sided ring and glucose has a 6-sided ring. So we'll be looking more at the structures and properties of our sugar units in session 11, which is our next session. So remember that I told you that the ether is a 2R group functional group, and it can actually be used to connect 2 smaller compounds and create a larger one. And this is how our small sugar units, single ones, can actually fuse together. So the example we've got here is where a single sugar unit glucose and a single sugar unit fructose actually fuse together to make a sugar called sucrose. So some of you have probably heard of sucrose. It's actually the sweetest sugar there is. Well, I guess technically fructose is sweeter than sucrose, but sucrose is our table sugar, and it's what you would commonly see as mentioned as sugar on your food packaging. So although people use the word sugar to sort of talk about carbohydrates in general, technically when you're talking about sugar, it's really sucrose. Okay, so what is sucrose? It's where we've got the glucose and fructose fused together through this ether linkage. And where we've got this ether linkage, it's also called a glycosidic bond. So where do you think the ether linkage is that connects the 2 sugar units within sucrose? Well, it's right here. So you can see that this oxygen between the 2 sugar units is actually bonded to one side using this bond, and is also bonded to the other sugar on the other side using that covalent attachment. So we've got an ether that is connecting these two, which we also call a glycosidic bond. So there's the ether, and the position of the ether, we also can say that there's a glycosidic bond here, which is basically attaching those 2 single units together. So we're going to look at a little bit more detail of how this reaction actually happens by looking at this short animation of a glycosidic bond. So I've drawn the OH groups that are going to be interacting here as oxygen bonded to a hydrogen. That's what an OH group is, but usually for simplicity we just show it as OH. Okay, so remember, in order for a new chemical bond to form, like the one between the oxygen in the middle here that forms that new ether, we need to break old bonds. So which bonds do you think are going to be broken within our 2 glucoses here that are going to attach together? Let's see if you can predict the right ones. It's going to be from this glucose, we've actually got the breakage of this bond here, and from this glucose, it's actually this bond here. So it's pretty tricky to predict, but essentially what's happening is we're losing the atoms that make up a water molecule, and they can come together to form water. So we've got our H2O up here. So let's look at what we're left with. One glucose now has an oxygen which needs an extra bond because it just lost a hydrogen, and this carbon here on the other glucose needs an extra bond too because it lost its attachment to its OH group. So what do you think is going to happen next to satisfy the bonding arrangements of those the carbon and oxygen that I've highlighted there? Well, we can get a new bond formed between the oxygen and the carbon, and that satisfies the bonding arrangements. So as long as they can move close enough together, then that is going to result in this formation of the new bond, and therefore the formation of the ether functional group, which has actually linked the two sugars together. And you can see there that we've actually now formed a glycosidic bond. So in green here, there's a bond, that's the bond that we've just formed, and we can call that the glycosidic bond. So not only do we call it an ether functional group, but a glycosidic bond. Glycosidic basically is just another word for sugar. So it's basically a bond between sugar units. So you can see from what we started with, which was two individual glucoses, we've been able to release a water molecule, which allowed the two sugar units to attach together by getting a new bond to form between an oxygen and a hydrogen between the two sugar units, creating the ether functional group and that new glycosidic bond. And you can see a short description of that on the right hand side. Alright, so now that we've gone through that content with the ethers, it's now a good opportunity to have a go at the first two questions. So as an online student, you can access these questions via the LMS under the weekly tab for this session. So I encourage you to have a go at those first two questions to confirm your understanding of what we've done so far. So pause the recording now and have a go at that. Alright, so to summarize this session, we saw that this part of the session, we saw that the hydrocarbon derivatives are compounds that contain atoms such as oxygen, nitrogen, and halogens, but also those compounds are mostly made up of carbon and hydrogen atoms. So where we've got a carbon and hydrogen atom compound, but the presence of some of these other atom types as well. So we saw all those ether compounds that had oxygen atoms attached at different positions, but most of the atoms were still the carbon and hydrogen atoms there. So some of the properties of ethers, so we saw that it was a 2R group functional group, and that meant that the functional group was going to be present in the middle of the compound, which allowed it to function to attach two components together, for example, two sugar units that we just saw connecting together. So to name the ether, we have oxy in the middle of the name, and we also had a look at an interesting example of how ethers were used as anesthetic agents, and the halogenated version of the ether was preferred because it had less side effects. So we also saw that the ether could be present in a cyclic ring compound where the oxygen atom connects to two carbon atoms that are both part of the same ring. And this was also the confirmation that we see in some carbohydrate units like glucose and fructose can exist in the ring form. And I also showed you how we can fuse together two individual sugar units like glucose and fructose to create a new sugar unit that contains two units. And to fuse those together, we've got the ether functional group in the middle, which corresponds to the position of the glycosidic bond, which is our sugar bond. So now we're going to change track a little bit, and the next part of the session is going to be looking at functional groups that contain the nitrogen atom. So none of the other functional groups that we've looked at so far contain the nitrogen atom. So we're going to spend a little bit of time as a lead-in to look at how the nitrogen atom can actually form its covalent bonds. So because the nitrogen atom is in group 5 of the periodic table, that means it has 5 valence electrons. So this is going to dictate how it's going to be bonding. And it's different to the bonding of oxygen and carbon because they have different numbers of valence electrons in their outer shell. So before we get on to the bonding arrangements, let's just do an overview of the amine functional group. So the amine functional group is essentially a nitrogen atom that is connected to 1, 2, or 3 R groups. So this is quite different to what we've seen with all the other functional groups we've looked at because the number of R groups attached is actually variable. And this is because the nitrogen atom can actually attach to hydrogen atoms, or if it doesn't have hydrogen atoms attached, that means an R group can be present in that position instead. So for example, this first version where we've got 2 hydrogens attached to the nitrogen, that means there's only room for 1 R group because each nitrogen atom forms 3 covalent bonds. But if the nitrogen atom within the amine group loses a hydrogen atom or has 1 less hydrogen atom, essentially there's more room for R groups. So we can see in this next example, the nitrogen has only 1 hydrogen attached, which means now that there's actually room for 2 R groups. And if there's no hydrogens attached, then that means all the positions bonding to the nitrogen can be filled with R groups. So they can attach to 3 different sets of atoms. So although all of these are considered the amine functional group, it can actually attach to different numbers of R groups. And I'll show you a way to actually designate what is a 1 R group, 2 R group, and 3 R group amine by using this nomenclature of primary, secondary, and tertiary. So we'll look at that a little bit later on. So the naming convention for a compound that contains the amine group is just to have amine at the end of the name. For example, propanamine. So prop tells us there'll be 3 carbons, and the amine tells us that there's the amine functional group. So here is propanamine. So where's the amine functional group? It's at the end here. In this case, it's got 2 attachments to hydrogen, so it's just the single R group, which is the 3 carbon chain on the left hand side here. So there's 3 carbons, which is the R group. So keep in mind, because the nitrogen can exist as either a 1, 2, or 3 R group functional group, this is going to have an effect on where the amine is present within a compound. If it's a 1 R group functional group, will it be found at the end or in the middle of a compound? Well, because it's only got the 1 R group attachment, it's going to be found at the end. Whereas the ones that have 2 and 3 R groups, would you expect those to be found at the end or in the middle? Well, these ones are actually going to be in the middle of a compound because they're going to have an R group attachment, which is a group of atoms or another atom type, on each side. Alright, so let's consider the bonding arrangements of nitrogen now. So if we look at the Lewis dot symbol representation, we've got a circle representing each of nitrogen's valence electrons. So remember I told you that the nitrogen was in group 5 in the periodic table, and that means it has 5 valence electrons. So because there's 4 positions within the outer valence shell that can take on electrons, 3 of these positions have an unpaired electron and 1 has a pair of electrons here. Remember, each position gets 1 electron before any of them are paired. So because there's 5 valence electrons, 1 of the electrons will actually get a partner. So it'll be a pair of electrons. So to work out how something's going to bond, what we really want to know about is the number of unpaired valence electrons. Because an atom will form bonds based on how many unpaired electrons need to become paired to become stable and satisfy the octet rule. All with the goal of getting it up to 8 valence electrons where all of the electrons are paired. So if we look at this Lewis dot symbol of nitrogen, we can see that there's actually 3 of our valence electrons that are unpaired, meaning it needs to form 3 covalent bonds. So you can see some different ways that the nitrogen can actually form these covalent bonds. In theory, it could be 3 single bonds, it could be a double bond and a single bond, or it could be a triple bond. So now would be a good time to pause the video and see if you can work out which atoms the nitrogen could bond to form 3 single bonds, a single and a double, and a triple. Remember that not only does nitrogen's bonding arrangements have to be satisfied, but so do the other atoms involved. So I'll give you some time now to pause and have a go at doing that. Alright, so keep in mind that what you've done might not be exactly the same as what I've done because there are lots of different combinations. So the most common thing for nitrogen to single bond to, just like carbon, would be a hydrogen. So in theory, each of these single bond positions could be taken up by hydrogen, and that would form the compound NH3, which is actually ammonia. What about if the nitrogen is bonded to something that needs a double bond? Well, oxygen forms a double bond, so we can put an oxygen there and then a hydrogen on the left-hand side here. And because nitrogen has 3 covalent bonds, it can form a triple bond. So a logical thing for it to triple bond to is another nitrogen, which would form the nitrogen molecule, N2. So to summarize what I've shown you here, we know that nitrogen belongs to group 5, meaning it has 5 valence electrons. So looking at the Lewis dot symbol of these 5 electrons, we see that 3 of these valence electrons are unpaired, meaning it needs to form 3 covalent bonds to get those 3 shared pairs to make it up to 8 valence electrons to satisfy that octet rule to give the atom stability. Alright, so now let's look at some animations which show how the nitrogen atom is actually going to form some covalent bonds with different atom types. So here the atom types, we've got the nitrogen, obviously, and we've got hydrogen. So how can nitrogen and hydrogen bond together? Well, because each hydrogen has a single unpaired valence electron, we can get an association between one of the unpaired electrons on nitrogen and each hydrogen. So once they come close enough together, we've now got this electron sharing relationship between the nitrogen and the hydrogens. So what do we call this electron sharing relationship that I've shown here? It's a covalent bond. So remember, in each covalent bond, we've got 2 electrons being shared between the atoms involved. So in this box here, we've got 2 shared electrons between the hydrogen on the right and the nitrogen on the left. So to satisfy the bonding arrangements in this case, the nitrogen will actually be forming 3 covalent bonds to 3 different hydrogen atoms. So all of the atoms involved here stable? All the nitrogen and the 3 hydrogens? Do they have the right number of electrons now? Yes, they do. Nitrogen has 8, so because these electrons are being shared, it now counts towards nitrogen's total. So 1, 2, 3, 4, 5, 6, 7, 8. And each hydrogen has 2 shared electrons, which gives it a full outer shell because it only has the 1 electron shell. So usually we represent this as the simplified representation where the line represents the covalent bond. So we've got those 3 attachments to hydrogen there, which, as I said earlier, NH3 is the compound ammonia. So this time we're going to look at a compound that can form between nitrogen, hydrogen, and oxygen. So the oxygen this time is forming a double covalent bond with the nitrogen atom. So you can see that 2 of the unpaired electrons of nitrogen are actually forming these covalent bonds to the oxygen. And the 3rd unpaired electron of nitrogen is bonding to hydrogen. So within that double covalent bond, it corresponds to 4 electrons being shared between the nitrogen and the oxygen. So to simplify this representation, we've got the nitroxyl compound where we've got a nitrogen double bonded to oxygen, shown by these two lines, and a single hydrogen atom as well. So remember I told you it can exist in these three different forms depending on how many R group attachments it has. So let's have a look at how we can designate between these forms. So the first type of amine you can have is called a primary amine. So the definition of a primary amine is where we've got a nitrogen atom within the compound that's bonded to only one carbon atom, which corresponds to one R group. So in this example structure, we can see that the nitrogen atom, which is part of the amine functional group, is attached to just that single carbon atom corresponding to a single R group. The other two attachments are to hydrogen and therefore not R groups. What about the secondary amine? So the definition for our secondary amine is if we've got a nitrogen atom bonded to two carbon atoms corresponding to having two R groups. So you can see in the example structure, if we look at that central nitrogen, it's actually attached to two carbons corresponding to the R groups. So they don't have to just be CH3s, they could be longer chains of carbons, but in this case I've just shown them as CH3s. And the other attachment is to a hydrogen. So we would designate this as a secondary carbon with two with a little circle on it. Whereas this structure here we would call a primary. So essentially you're just looking for the number of attachments to carbon from that nitrogen atom, and that corresponds to whether it's primary, 1, secondary 2, and the third option is a tertiary amine. And this is where the nitrogen atom has all of its bonding positions taken up by bonding to carbon. So there's 3 attachments to the carbon atom, which we can see in the structure down the bottom here. So we've got the nitrogen atom in the center there, and every single one of its covalent bonds is to a nitrogen atom, corresponding to 3 different R groups. So you can see in a tertiary carbon, a tertiary amine, we've got no attachments to hydrogen. All of the attachments are to carbon instead. So I'm going to take you through how to work out whether your particular compound that you're looking at is an amine that is classified as primary, secondary, or tertiary. So you'll have a go at doing some of these yourself, and there's also some in the revision questions as well. So the first thing to do, step 1, is to identify where the nitrogen atom is in your compound, because that's the position of the amine group. Okay, so where is the nitrogen atom? Well, we've got the nitrogen atom position shown with this red circle around it here. So that's the position of our nitrogen atom. And step number 2 is actually to work out how many carbon atoms are attached to that nitrogen atom you've identified in step 1. So the nitrogen atom was in the center there, and you can see the attachments to carbon have these blue circles around them. So there's 2. And the third step is to recognize that the number of carbons identified in step 2 specifies whether the amine is primary, secondary, or tertiary. So if there was 1 carbon attached, that specifies primary amine, 2 carbons attached, specifying secondary amine, and the third option being 3 carbons attached to that nitrogen, being a tertiary amine. So in this case of the structure we were looking at, the compound had 2 carbons attached to the nitrogen, and therefore we can call it a secondary amine.